All right, so thanks everyone for joining. I'm going to give so my name is Mathieu Chapira. I'm a prof at U of T, at University of Toronto, sorry, and I'm overseeing this Dream Cash Target 2035 challenge. So I'll give a brief introduction, and then Luca Chiesa, who is really driving this, will give us a more detailed overview of the challenge and answer questions that you may have. We also have on the call some of the folks who run the Dell screen for more technical questions, if any. Yeah, so let's get started. So this is the second Target 2035 Dell ML Challenge. So Target 2035 is a open science initiative with the goal of developing potent, selective, and cell-active chemical modulators for all for as many protein in the druggable in the human proteome as possible, at least those that are druggable. Yeah, so this challenge is composed of several steps. The first step is a retrospective. So the goal, sorry, I should start with this. The goal is to train machine learning model on DNA encoded library screening data. So it's large, rather noisy data. And then the goal is to use the model that has been trained to predict bioactive drug-like molecules. So drug-like molecules that bind the protein target of interest. So the first step is a retrospective step. It's ongoing now, but it's definitely not too late to start. You have until September 15 to make your predictions. So here you're going to predict ligands from that small molecule ligand known to us but hidden, and then we'll see. And the leaderboard will and Luca will tell you more about this. And then the third step, for those who do well in the first or the second, is a prospective step where you use your machine learning models that have been successful in step 1 or 2. You use this model to predict compounds from the enamine, so commercially available molecules, and then we'll target of interest to the Gates Foundation for contraception. And if you are interested, you can read more about this. The training set, so it's a Dell screen on 890 million molecules, for which you have the SMILE strings, the sequence read counts, so which correspond, if you will, to the binding signal, building block composition of each molecule, and this screen was conducted in the lab of Damian Young, and really this exercise was led by Kevin McKenzie, who is on the call here, at the Baylor College of Medicine. And when I say one screen, I should say three. There were three. You have three Dell screens, one against the apoprotein, so just the protein by itself, one against the protein plus a known ATP competitor, so a small molecule that binds to the ATP site. And finally, a third screen where it's just the bits, no protein. It's a negative control. So in principle, the compounds that are of interest are the ones that show up in the first bullet point, but neither on the second nor the third. And that's important. We'll come back to this in a moment. And the test set is composed of 400,000 compounds that were screened at the Structural Genomics Consortium by Affinity Selection Mass Spectrometry, or ASMS, which is another binding assay, just like Dell is a binding assay. And this was conducted in the labs of Huipeng and Levon Halabilyan, and Sunny was really driving this. And out of the hit candidates from this ASMS screen, 450 were confirmed in an orthogonal assay, a kinase ADP-Clo assay conducted at INSTEM in India in the group of Neil Kamaria, who is very much focused on PGK2. And so this test set of 400K compounds is actually split into two, so that in the leaderboard you can make prediction on one split or the other. And Luca will come back to this in a moment as well. And last slide before I hand the stage to Luca. Just a word of caution here. This is a structure, 3D structure of the protein target PGK2. So this is the site where non-ligands are binding. There are a few non-ligands. They are binding on this site. This is where the ATP binds as well. And so this kinase catalyzes the transfer of a phosphate from here to the ATP to the substrate, which is a phosphoglycerate. But you see that there are other pockets in this protein. And probably, I'm not sure, I don't know, but probably the DEL data contains compounds that bind here a lot, and maybe some that bind here, some that bind there. And similarly, the DEL data, the ASMS data would include compounds that bind here, or there. So, but the good thing is that both you have data sets that are focused on the catalytic side, because the DEL data, as I mentioned, was conducted against 3 version of the protein, where this binding site is available, one where this binding site in principle is occupied by a small molecule ligand, and one with no protein at all. So you want to focus on the compound that bind in the protein where there's in the apostate, so without a competitor. So that's the DEL ATP focused DEL data. And the test set, again, the ASMS in principle is pocket agnostic. Oops. But it was the compound, the 450 true positives were confirmed in a kinase assay. So in principle, they should also bind here, or they should be enriched in compound who bind that do bind here. So both the DEL and the ASMS data should be enriched in compound that bind here if you use the data in the right way. And I'm going to stop sharing here. And yeah, give it over to Luca. Oh, and questions. Sorry. So for questions, I guess you can use the Luca. Do you want to tell people how to when and how to ask questions?
Yes. So if you have any question during the presentation, you can write them in the chat in the meantime, so you don't forget. And then at the end of the presentation, you will have some time to answer any question. It's from a logistical point of view, it's easy if you just write your question in the chat, so we can also group together similar question. So okay, so more in detail, more on the practical details. For the challenge, as Matthew presented, so the challenge was launched on June 15th, and the retrospective part will run until, October 15th. The challenge is hosted on the Synapse platform from Sage Bionetworks, so you can, for people that participate in the previous challenge, it's the same website, and all our data is hosted on the IRCheck platform, which is a platform that was developed for the Target 2035 initiative to host ASMS and DEL screening data. As a small note, for people that participated in the previous challenge, this one will be a bit different because we listened to the feedback you gave about it. So we have a few updates. So now all data in the challenge have smiles for the molecules. So the structures of all the molecules in the dataset is known. You will get immediate feedback after your submission. So you will, once you submit your prediction, you will get in a few minutes your results. The challenge, as you see, is not 1 month anymore, but to run for 3 months, and also we added some extra information about the data since we understood it's not everyone is a DEL expert outside. So more in detail. So as Matteo said, the challenge will be split in 2 main phases. We have a retrospective phase, which is open to everyone. Everyone can participate. And then we have a prospective phase, which instead is reserved for the best performing groups. So the retrospective phase itself is split in 2 parts. In the first part, we call it the blind test or the blind hit discovery. You will be tasked to find 431 hits identified from an ASMS library and confirmed in an enzymatic assay. The data is split between a validation set and a test set. So the validation set was prepared because we know that it's difficult to create the perfect workflow on the first trial. So we decided to have this data set apart so you can make up to 100 submissions during the entire challenge. So you can test your workflow and see how well it performs on unseen data. Each submission will be formed by 50 molecules. So you have to write the catalog ID of the molecules as shown here. You submit the list and you will get an immediate feedback on the number of hits you identified and the number of chemical series. On the test set, instead, you have only 2 submissions. So you have to be very careful on which workflow you want to use. And in this case, you have to do 2 things. One, you have to score the entire test set because we will also evaluate you based on the ranking power of your method. And also you have to select 50 molecules for evaluation. So the objective of the challenge is to find as many diverse hits in the best 50 molecules your workflow can identify. Okay. After this phase of the challenge, we will release all the hits that have been identified in the test set by any group. So if of the 200, around 200 hits that are in test set, all the groups identify 50, will reveal all 50 molecules, and you will have an additional month, so from September 15th to October 15th, to refine your workflow, tune your model, and try to find even more hits from this data set. Again, you still have 2 submissions on the test set because we want to give you the opportunity, if the first submission does not go very well, to adjust and reorganize, but also you have another 100 submission on the validation set, so you can try and refine your model. Very important, this is a DEL-ML challenge. So we want to see how good machine learning models trained on DEL data are. So for a workflow to be considered for the challenge, it has to include the use of DEL-ML. This does not mean that you can only use DEL-ML in the sense that you can use other tools inside your workflow. So for example, you want to use the machine learning model to go from 200,000 molecules to 1,000 molecules and then do docking and FEP on all 1,000 molecules. That's perfectly fine for us. You want to dock all the molecules, use the docking score together with the DEL-ML score. This is also fine for us. Don't just dock all molecules in the test set because it goes against the spirit of the challenge itself. So, and this is what concerns the retrospective part. After this, there will be a prospective part for the best performing groups. So for the 5 best performing groups in the blind test set and in the active learning step, will be invited to take part in the prospective phase, in which we'll be asked to screen the Enamine US liquid stock collection, which is formed by around 2.7 million molecules. This part will run for a month and a half, so from October 15th to the end of November. And as during the retrospective phase, each group will be tasked to select 50 molecules to submit, and this molecule will be tested experimentally. As a small note, so we will remind it to the groups that will be invited, before purchase, we will test the molecules ourselves computationally to filter for solubility, possible solubility issues, and for molecular novelty, because we want to be sure that we are trying to discover novel ligand, not just analogs of ASMS is. And one important thing, so participation in the retrospective phase is free. Everyone can participate. You are more than welcome. In the prospective phase, participation is not free. SGC will cover all the experimental costs, but you have to purchase the compounds using your own funding. As an upside, though, SGC will cover the cost for purchasing compounds for the best two mainframe teams in both steps. So the best two team in the blind test step and in the active learning step will cover the purchase of the compounds if they are part of mainframe, which means that you will make your workflow open source and available for everyone. So if your model is closed source, you can still participate in the prospective phase, but you'll have to cover the purchase of your compounds. Additionally, since this time we are partnering with Cash Challenges and Conscience to run the full challenge, there is additional funding and invitation slots for Canadian participants. So if your group is from Canada or some members of your team are from Canada, I invite you to contact Conscience and they will give you all the details about how to access this additional funding. Finally, for everyone, at the end of the challenge, we'll release all the data. This is an open science challenge, so all the data will be made public and no patents will be filed on the discovered molecules. And at the end, we will write an overview paper to describe the results of the challenge and the main methodological trend we observed. As you can see here in the slide, we already released a preprint for the results from the first challenge that you can read on ChemArchive. To be eligible to be invited as a co-author, you need to satisfy two conditions. One, your model needs to perform better than random selection. This is to reward groups that are not only put the work, but also were able to build something that works. And also you need to provide a satisfactory write-up describing your workflow. So to be considered for the overview article, we need to know what you did and have an idea of how your workflow works. As a very important, the write-ups needs to give sufficient detail that an expert can learn something from it. It's not mandatory to make your model or your workflow fully open source for be considered for co-authorship. So if you are a startup, you are a company, you don't want to make your model open source or you don't want to share too many details, it's fine for us. You have to give us some details. You can see in the example, if you just write, we used our proprietary model to predict ligand binding, that's not sufficient for us because it does not give us any information. Instead, if you tell us we used our proprietary model, which is an attention-based graph convolutional neural network trained on DEL data, then at that point, it's closer because it gives us an idea of what works and what doesn't. Okay. Again, you don't need to make your code open source. We will really appreciate if you will, but it's not mandatory. So other important information. So as I said at the beginning, all the data is available on ArCheck, which is a platform. You can scan the QR code here in the presentation to go directly to the web page. Important, this dataset that you are going to download is quite large. It's, if I remember, a few gigabytes, a bit less than 10, I don't remember exactly. So it's not open to download for everyone. To download it, you have to register to the ArCheck platform. It's very easy. You just have to go to the web page. There is a login button. You click, you can decide if you want to log in using a Google account or with your email. And after this, you need to request authorization to download the datasets. It's not complicated. You just have to go to view profile and then go to apply for datasets. You just have to fill the form and then you will be authorized. Important, authorization will be accepted automatically if you are a mainframe member. So if you use to register to ArCheck the same email you used for mainframe, you'll be accepted automatically. If you register using an academic email, you will probably be automatically accepted. If instead you use your Gmail, it's fine. The only thing is that in this case, you'll need to be manually accepted, and this might take some time depending on our availability. So factor this while preparing your work. So a bit more on the Dell data, since as Matt already mentioned, you have access to the full data on the Baylor College of Medicine Open Dell library, which contains more than 800 million molecules, and we have a free set of screening, PGK2 alone, PGK2 pre-incubated with an orthosteric ligand, and a control for possible interface with the matrix. As I said, as already mentioned, you already know we want molecules that bind the ATP binding site, not that bind allosteric sites. So in the test set, active molecules are ones that bind either the ATP site or the cofactor site. Very important, unlike the previous challenge, we are not giving labels to the dataset in the sense that when you download the dataset, we do not tell you which molecules are considered as active from the Dell screening and which are considered inactive, because we don't want to add this. Since this is a calculated information, we don't want to give it to you. If you're not an expert, we have instructions on the wiki on how we recommend to label your data. So no need to panic. It's very easy, very simple, quite quick to calculate. Most of the dataset preparation has been already handled by us, or to be more specific, by our team at the University of North Carolina that has done all the dirty work, and by the people at Archak, of course. We leave the finer details on the dataset up to you. In the sense that you have 800 million molecules, you might not want to use all of them. We leave up to you which molecules you want to put in your training set and which one you want to leave out, for example. For the evaluation, so you'll be evaluated based on 4 criteria based on importance. The number one is the number of chemically distinct hit series that you will find in the 50 selected molecules. So we already pre-clustered all the active molecules. Each molecule is assigned to a cluster and will count the number of unique clusters you identify. After, if 2 groups are tied by the number of chemical clusters, we will move on to the total number of hits. And then if teams are tied on the number of hits, we will evaluate based on precision recall curve AUC on the full library and on ROC AUC on the full library. As a personal note, which might be wrong, because again, this is very difficult for us to, it's not a generic trend, but based on what we saw from the previous challenge and some internal testing, chemical diversity is included to cover corner cases because there are some hit molecules that are very similar to each other, and might be that some algorithm might find a lot of hits, but these hits are very similar to each other. So the reason why we account for chemical diversity is to cover this corner case. Based on the results, identifying hits is still the bottleneck in the sense that do not think, yes, I will find 20 hits out of 50. I have to maximize this hit rate. The problem will be, yes, I might not find hits. You can already see some of the results on the validation set. I think the group that found the most hits has found 3 hits that cover 2 chemical series. So again, chemical novelty is important. Don't focus too much of it in the early stages. First, try to optimize the number of hits, then you can optimize for chemical novelty. So other important things, because there are a lot of information, I'm sorry for the text wall. So number one, Dell data are noisy due to the nature of the assay itself. The labeling in the training set might not be correct in the sense that if you take a molecule from Dell, you resynthesize it and you test it, a molecule that was flagged as active in the assay might be inactive, and vice versa. Something that was flagged as inactive might be active. So be very careful, be very mindful of this. Both training and test sets are imbalanced. So we have way more inactive molecules than active molecules. So again, something to be mindful of. As I said, both data sets are pocket agnostic. I think we covered this quite well already. And another very important thing is that there is very little overlap between the chemical spaces of the training set and the test set. So this is a mostly out of distribution challenge. So it will be quite difficult to get a very good model. So be creative, try your best, try different approaches, because it will not be a straightforward, I train, I get very good results. We did some preliminary studies on this dataset. So baseline LightGBM model trained on ECFP4 using all the deletes and a subsample of the inactive was able to find 5 hits that correspond to 3 chemical series in the validation set, and it was able to find 1 hit in the top 50 ranked molecules in the test split. So it's possible, baseline models can perform well, so it's not an impossible challenge. Again, the results are quite limited. So again, it will be up to you to try and improve on these results. As a side note, we also tried some models from IBM called Mammal and Melon. We got some interesting preliminary positive results. These models are foundational models that can be fine-tuned using data. On the wiki page, we put a link on a tutorial on how to fine-tune them. So we got some preliminary results. Again, they might not be the best models. We cannot guarantee that. So you are more than welcome to try them and tell us if they are the best solution. And last thing, very important, the way we built the validation and the test set is we build them by scaffold splits. So there is very little scaffold overlap between the two. So a good performance on the validation split does not guarantee a good performance on the test split. So be mindful of this. A model that finds 20 hits out of 50 on the validation split might still find 0 hits on the test split. So be careful to not overfit your model on the validation set. And finally, last thing is on the deadlines and the write-ups. So all submission deadlines are based on UTC time. So please do not wait for the last moment for your submission. So don't wait until the 15th of September to send your submission. Please try to send them by the 14th. It's a bit better. We require for ease of evaluation that for each submission, you send a separate write-up. And we have set up a write-up template that you have to follow to describe your model. This is done mostly because, as we saw from the previous challenge, a free template gives us some problems in finding all the key information. All write-ups are accepted until the end of the prospective phase. So if you don't want to share your method right away, you want to wait until the end of the challenge because you are afraid someone will steal it, you can wait until the last day. And as a small thing, as the ChemSpace team from the last challenge told us, one of the most difficult things for them was to wrap up all the experiments to write the final write-up. So please do not wait for the last moment to document your workflow. It will be better for all of us. Okay, so finally, I invite you to register on the Synapse webpage. You can scan the QR code here. Now we'll have time to answer question, but this is not the only occasion in which you can ask question. If you go on the webpage, there is a discussion section. It works like a forum, so you can create your threads. The organizers read this forum relatively regularly, so we will answer you and we will give you all the information you need. And finally, I want to thank all the people that worked on this, both from the challenge organization, the people that contributed the data, the people that prepared the data and managed the platform. So thank you very much for the attention. I will leave this so people want to scan it. Okay. Okay, this is all for me.
Great, thank you, Luca. So there are a few questions. I answered some, not all. So So there was a question on especially on the definition of what count as novel.
Novel in which sense?
Chemically novel. You said
So for the retrospective phase, novelty is not a problem in the sense we perform the clustering of the active molecules to find chemical series. This clustering is not fingerprint based. It's uses a algorithm which is based on maximum common substructure. If you want more details on how it was done and how the test validation split was done, it's available on the wiki since it's not the most straightforward explanation.
I put a link to the paper on the chat box.
Thanks. And for the prospective phase, we will evaluate similarity to known ligands and to all the molecules that we reveal in the active step phase. I think for now the threshold we are settling is it needs to have Tanimoto similarity on ECFP4 less than 0.35. Correct me if I'm wrong. But again, this one will once we arrive to the retrospective phase, we will give you more details and we'll also interact better. I think it will also depend on how many molecules you find during the active phase step. If you find all 400, it will be, we might need to relax some limits, but we'll see on this. But ideally 0.35 ECFP4 similarity, Tanimoto similarity is the point.
Okay. Is there other burning questions that were not addressed yet?
Let's see. Will recording be shared after the meeting? Yes, we will load it on the signups page, so you can get this information back at any moment.
All right. Someone is asking for detail on whether and how to use docking. Do you want to take it, Luca? Or
1 second, can I see the question? So docking can be used. It's not a problem for us if you use docking. The point for us is that this is a DLML challenge, or it's a drug discovery challenge based on DLML. So if you just use docking, that's not the object, not what we want to achieve with this challenge. What we want to achieve is to use the data from DEL. Of course, if you say we used docking to rank the molecules and then we used DLML to get a second rank and we do an ensemble of the two, it's fine. If you decide to not dock all 200,000 molecules because it's too many and most of them are inactive and you want to use the machine learning model to whittle down the selection to the last 1,000, that's fine for us. It's more that DEL data needs to play a role in this process at some point. DEL data and hopefully machine learning in some form needs to play a role in this process. If you have doubts, you can always write in the discussion in the sense that theoretically, if you want to fine-tune bolts based on DEL data, I don't see why that's not a good idea. That one, I think we can consider it acceptable, for example, if you can do it. So again, it needs to use the DEL data somehow.
Yes, if you want to see the lessons learned from the first DLML challenge that did include many people who combined DLML with docking, I put a link in the chat box. You can have a look there.
Can you explain the process of creating a team? Okay, this is up to you in the sense that I guess, so I know some of the groups are already some labs, so they are already academic teams that are already formed. If you want to form a team with other participants, you can use the discussion session. I guess something important, discussion session, the most useful part is to use it to ask us questions. You can also use it to discuss between you. So if you are a single participant and you would really like to form a team with someone, you can start a thread in the discussion like teammates looking for teammates, and you can see if other people want to join you. Again, you can participate alone, you can participate as a team. In the last challenge, we had a few people that participated on their own. They got good results. Some people that participated as a lab, they got good results. Some people from different institutions that collaborated together, even outside an academic environment, and they still got interesting results. So that's up to you.
We use PGK2 from data from Campbell. Yes, any data that's public is.
Yeah, yes, you can use them. There are not a lot of data on PGK2 on Campbell, so if you find, yes, also I forgot to mention it because I forgot to put it in the slide. At the same time, there is a cache challenge running on PGK2. So if you go to the cache website, is on the wiki. This one is information you can find. There are some extra information about PGK2 from this cache challenge. You can easily access them, and you can use all of those information. I think at the moment we have more PGK1 ligands than PGK2 ligands, or they are like promiscuous between the two. You can use any information. Since we cannot stop you from using extra data are allowed. Still, you need to use DEL data in your models.
Yeah. And in your method description, we do ask you to please specify whether you augmented the training data set and how you did so. Thank you.
Okay.
Real quick, sorry, Luca, I just want to confirm the actual files for the challenge are thrown up onto AirCheck. Is that correct?
Yes. Yes.
Thank you.
Yes, but in general, like all the information that I gave you today and some more technical details that I did not mention today can be found on the wiki page. It's quite extensive. We tell you where data is, how to download it, etc. Again, if something is not clear, you can write in the discussion and we will promptly give you an answer on. If you can't find the data, we'll put the link back again.
So there are a couple of folks who are asking whether this presentation will be available online.
Yes, I will upload it to the Synapse page so you can access it.
Okay. Any final question, anyone?
Or equal are the libraries pulled in equal concentration or equal copy number? I can't answer. Maybe Jonathan or Seth can give you a better answer.
I actually think this might be a better question for Kevin, if I'm not mistaken.
The concentration question?
Yeah.
Equal copy number question? Yeah. So when the experiment was performed, each of the approximately 1 billion compounds was present in each tube at 1 million copies. So 1 million individual molecules is a starting concentration. And they should be, if our chemistry is good, in equal concentrations. When the library was made, some of the steps may not have been as efficient as others. No chemistry is 100%. So they might be off from that factor of 1 by 0.8, right? But they are in approximately the same starting concentration for every individual molecule.
Right. Thanks, Kevin. Okay. All right. So if any other questions, again, so the discussion panel on the Synapse interface is open, and we'll make sure to answer questions there as well. So this session will be available. So this webinar will be available for later viewing. Thanks everyone for joining. We hope to see you on the challenge, and we hope you make great predictions. Thanks, Luca, Kevin, everyone who joined, and have a great day. Bye-bye.